So welcome back, folks. This is part 2 of chapter 5. And here we'll start talking about the receptors in detail. So the first receptor to talk about are the ligand-gated ion channels. And we'll discuss, or I'd like to talk first about the structure of the receptor. So you see that the receptor is a transmembrane protein. So again, here is your inside of the cell, that's your outside, and there's your plasma membrane. There is your transmembrane receptor. That's where the first messenger is going to attach to. So usually the ion channel would be closed, okay? So until your first messenger comes and attaches to the receptor binding site, and that leads to the conformational changes of the third, of the tertiary structure of the protein, leading to opening of the ion channel. Now, ions are able to get in or out, okay, depending on which ion channel you opened, and that would lead to your cell response. Okay, so let's say if this had been a skeletal muscle, for example, calcium might have led to a muscle contraction. Okay, if this was maybe a sodium channel, it could have led to an action potential if it was a nerve, and so on. So you see here that there are, different ion channels.
The sodium channel has receptor sites
on which cell that we're talking about. And we'll like to watch this little tiny video in order to illustrate what we just discussed.
For a ligand acetylcholine. When the receptor sites are not occupied by acetylcholine, the sodium channel remains closed. When acetylcholine binds to its receptors, the channel opens. Therefore, the channel is called a ligand-gated ion channel. When the channel opens, sodium ions diffuse through and enter the cell.
Okay, so you see the scenario here with sodium channels. Right here, we've got calcium channels. And as I mentioned, there are so many different ion channels depending on where you are, and all of them will lead to different responses. So this is what we talked about in writing. Okay, so first off, the first messenger or the ligand would have to attach to the receptor, leading to conformational changes, changing again your tertiary structure of the protein, and that is going to open up the channel. Ion channels are kind of like a gate. Okay, so we have to open up that gate in order to allow ions to go through. Hence, we call them ligand-gated ion channels, since the key to open this gate is the attachment to a chemical. When the ligand-gated ion channels open, that is going to allow the diffusion of ions across the cell membrane, and that would lead to a response in the cell. Usually it is in the form of the change in the membrane potential of the cell. Now, the second receptor to discuss is what is a receptor that has an intrinsic kinase activity. So remember, we said our first kind of receptor is the receptor that doubles up as part receptor, part ion channel. Your second type is part receptor, part enzyme. And again, I would like to just talk first about the structure of the receptor before we talk about how it works. So you've got your receptor with the receptor binding site, and built in the actual receptor, there is a kinase enzyme. Remember, we said that kinases are going to add phosphates and they activate different proteins. Okay, the kinase is going to phosphate or activate an amino acid residue known as tyrosine. And when we do that, we have now activated the receptor. Now that receptor is going to activate other proteins. These other proteins are known as docking proteins that are in close proximity to the receptor but are not part of the receptor. So these docking proteins are going to be activated. So you see here we're adding again these phosphates. And these now activated proteins will lead to the cellular response. So you'll see this will become more of a a pattern with these receptors where it kind of has a domino effect. We activate the receptor activates other proteins, and it keeps on going on to lead, and that is really your cellular response. Okay, so at the end of all of this protein activation, something is going to happen in the cell. Either maybe we're activating Krebs cycle, or we're activating, some other kind of metabolic pathway, but that would be your cell response to your first messenger. Okay, so let's take a look at this video to kind of illustrate what we talked about.
Receptor tyrosine kinases exist as monomers free-floating in the cell membrane. Each receptor has an extracellular domain and an intracellular domain. When growth factor is present, it binds and induces dimerization of receptor monomers, resulting in receptor activation.
The dimerization is the change in the tertiary structure of that receptor.
The receptor then autophosphorylates tyrosines in its intracellular domain. The phosphotyrosines are binding sites for intracellular signaling proteins, which in turn can activate other downstream proteins. An exponential signal amplification occurs for each growth factor molecule binding the receptor tyrosine kinase.
So these receptors, as we mentioned, have that intrinsic kinase enzyme activity. The majority of the receptors are going to activate tyrosine residues, and they are known as tyrosine kinases. Okay, and again, your typical sequence of events would be that the ligand binds to the receptor, okay, leading to change in the structure of the receptor and its activation. And then the tyrosine kinase is going to phosphorylate. Did I skip something? I did. It will phosphorylate its own tyrosine residue. Now that newly created phosphotyrosine that's on the cytoplasmic side of the receptor is going to start activating or phosphorylating those docking proteins. Now these docking proteins are going to start activating other proteins downstream. And again, it's kind of like this domino effect that we are going to be, you'll see it as a theme within the net, pretty much the other receptors that we'll be talking about. Okay, so they are all involved in the activation of cytoplasmic proteins by phosphorylation. Now, we did mention that usually the intrinsic enzyme is a tyrosine kinase, but there are, just like in science, you should know this by now, that there are always exceptions. Sometimes the intrinsic protein is not tyrosine kinase, but it is rather guanylyl cyclase enzyme, and that is going to catalyze the formation of a molecule known as cyclic GMP. And this guanylyl cyclase leading to cyclic GMP is known now as a second messenger, okay, and that is going to activate other proteins downstream. This is very important, especially in the retina. Okay, it processes visual inputs. In the text, it goes into detail about how these visual inputs are processed. It could get very confusing on how the turn off, turn on kind of works. So focus on what we talked about here in this lecture. Now, the third kind of receptor that we need to discuss is a receptor that does not have a built-in kinase, but it is in very close proximity to one. So the janus kinase is the enzyme that is going to start the activation process, but it is found inside of the cell. So it is cytosolic janus kinase. So you see here a receptor that interacts with cytoplasmic janus kinases. So it's a little bit different than what we just talked about. In the previous receptor, we said that the protein kinase is actually part of the receptor, but in this case, the protein kinase is not, okay? So again, the same sequence, the first messenger combines to the receptor, changing its conformation, leading to activation of that receptor. Now that receptor is going to activate the janus kinase, which will lead to phosphorylation and activation of different proteins on the inside of the cell. And again, that will lead to your cellular response. These janus kinases, so this is what we just talked about, but in writing. Janus kinases usually end up leading to synthesis of new proteins. So although there is no direct interaction between the first messenger and the nucleus, okay, but your end result at the end of this cellular response cascade, usually there is a protein that is being synthesized. These are really important signaling messengers, especially they're known as cytokines, and these cytokines are made by our white blood cells, so they're basically really important in the function of our immune system. Okay, so this concludes part 2. I will see you in a bit in part 3.